but they're not so diverse. So while, yes, you could, for example, try to use polysaccharides as an immunogen. They have to be pretty high molecular weight. You'd want to have a lot of different kinds of sugars in it, maybe modify those sugars a whole lot, and then it would possibly work as an immunogen. But generally speaking, sugar polymers don't work very well. And the same goes for nucleic acids. Lipids, on the other hand, have very little diversity among them. And although they can be cut, it's generally a much more laborious process for the cell, the body. So that doesn't work very well. Drug species usually are too low molecular weight, and plastics, both not very diverse and really hard to degrade. So we don't generally show any, we don't show an allergic response to Teflon, for example. All right, so we've got vocabulary. We've talked about the FAV, FC, heavy chains, light chains. Here's some more vocabulary. Epitopes and haptens. What's the difference here? Well, the epitope responds to a particular set of molecular features, like the side chains of amino acids on a polypeptide. That would form an epitope. A hapten, on the other hand, is like a complete compound, usually low molecular weight. So if you give somebody, sulfonamide, that would be something that might not elicit an immune response. How could you get antibodies to sulfonamide? Well, what you could do is cross-link it onto so-called carrier protein. Now it's part of a protein that has a lot of diversity, and it's just got some added diversity because of the chemical features of your sulfonamide. Now you've got a chance for it to be cut up, processed, and bound, fragments containing the sulfonamide, and then you get an immune system response to that. That's what happens a lot of times, in fact, with drugs that are low molecular weight, and people show an allergic response. The drug gets cross-linked onto some of your native protein, then that gets recognized by the immune system as a weird protein that didn't have drugs on it. Danger, Will Robinson. Time to cut that up. And it processes that in your immune system, and so you end up with an allergic response. So short peptides, usually not immunogenic, but if you link them onto a carrier protein, so if you got a small hormone, probably not going to work. Melatonin, okay? Use that as a sleep aid. Generally, people are not allergic to that. But if you cross-link it onto some sort of a foreign protein, yeah, then it becomes immunogenic. So the same thing for these drug molecules here. We generally have to conjugate them onto a carrier protein in order to get an immune response. So if we're trying to make monoclonal antibodies to drugs, are you going to just feed the drug to the animal and hope that you get the right kind of antibodies? No, probably not. You're going to have to do some chemistry beforehand. You're probably going to have to cross-link that drug onto your carrier protein. All right, so here's a series of compounds, and I'd like to go around the room here and around the circle here, and let's discuss which of these would be immunogenic by themselves and which wouldn't. And if they're not, how could you get an antibody to it? Let's start up here with Celebrex. Here's a sulfonamide moiety. Here's, let's see, CF3 and CH3, benzene rings, okay? Is that going to be a very good immunogen? No, why not? Low molecular weight. Does it have a diverse set of features? Sort of not. Yeah, sulfonamide is kind of unusual for most biochemical systems. It's hard to find those. Those are usually man-made. But benzene rings show up all the time. So do methyl groups. CF3 looks an awful lot like a methyl group. It's just about as greasy. So it's probably not going to be very immunogenic either. So overall, this probably is not going to show much immunogenicity. Too low molecular weight, not enough difference here. Now let's go over to this. This is an antibiotic, gramicidin S. It operates by creating pores in membranes and letting protons and small ions through. And that destroys the electrochemical gradient across the membrane, and that blocks the cells from making energy. Is that going to be a good immunogen? We see some nodding heads in Chicago. Let me hear from Rockford. What's the consensus from Rockford about this?
Not sure if it's big enough, but
Not sure if it's big enough. Okay.
there's some like D amino acids which aren't very common.
So we've got D-phenylalanine. We've also got this unusual amino acid ornithine showing up here as well. So that's kind of unusual to see that polymerized. So your body might recognize that, or maybe the D isomer here. It's also a polypeptide. Remember one of the other things that we require? You have to be able to process this. You have to be able to cut it up so that your antigen presenting cells can display pieces, parts of it on their surface. Couldn't really do that with Celebrex. But yeah, peptidases can go to work and cleave those peptide bonds between these amino acids. So this could be a potential immunogen. Well, let's go down here to Dursban. Anybody recognize that? Nobody's worked on a farm. It's an insecticide. Okay. Yeah. It's a neurotoxin, in fact. This thing will cross-link onto acetylcholinesterase and block it and kill off the nasty little insects that are trying to eat up your crops. Is this going to be a good immunogen? No. Why not? It's too small. Good choice. Can we break it apart? Well, yes, we could. I can see some stuff that could be hydrolyzed here. But there's not enough diversity here, and it's really small. If we hooked it onto a protein some way, possibly then we could get an immune response to it. But all by itself, you're probably not going to get an allergic response to it. I don't recommend breathing or eating it, however, because, like I said, it'll go after acetylcholinesterase, so probably not a very good thing to put on your shake over your diet. Okay. What about this polymer? It's a polymer. We want high molecular weight. Can we hydrolyze it? Why, yes, we could. Glucosidases will chop that right up.
I don't think it would be because it's glycogen, and that's a very common thing to see in the body. So hopefully your body doesn't amount to an immune response to it.
Excellent response. Yeah. What are the monomers here? They're all the same. There's one more reason why. They're all glucose. Hopefully you're not allergic to glucose. All right, last one. Anybody recognize what this thing is? Well, yeah, it's peptidoglycan from the bacterial cell wall. Now, would that be a good immunogen? Let's have a vote. I mean, democratic decisions here. That sounds like the thing to do. So how many think it's a good immunogen? And how many say, no, 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 no? And how many are not going to raise their hands? Yeah. Okay. All right, first, do we have high molecular weight? Yep. Do we have chemical diversity? Yep. Are those features unique? That is, we don't see a lot of that kind of stuff in our body. Now it gets interesting. Okay, we've got sugar hooked up to peptide. And we look over here and, oh, look, D, glutamic acid, D, alanine.
Okay.
We don't usually see that in our body. Those are foreign-looking things. Yeah, this thing would probably work as an immunogen. We can chop it up. It's high molecular weight. It's got lots of different kinds of chemical features, and they're features that we don't normally see.
Yeah.
So, everybody getting the idea how to treat these kinds of problems now? Okay. All right, so monoclonal antibodies. So these are antibodies that you end up producing that are all identical. They have exactly the same kinds of heavy chains, same kinds of light chains, same kind of antigen binding site, same specificity for the same antigen. You can produce them in a large volume, and you can keep on producing them as long as you take care of your cells. So all of that makes it very useful for developing all sorts of assays and treatments. Monoclonal antibodies are often used in treating certain kinds of diseases, and you'll again learn way more about that next year in 504. But there are disadvantages to these. First of all, it takes a long time to go through this whole process to end up with an antibody for whatever compound you're interested in analyzing. And because that takes a long time and it takes a lot of materials, animals, cell medium, growing in very strictly controlled conditions, avoiding any mycoplasmas and so on and so forth, very costly. And then I put down as one last disadvantage, detects a single epitope. Wait a minute, didn't I say that was an advantage up above? Well, we usually think of detecting that single epitope as the major advantage. They're highly specific in what they bind. But because they detect a single epitope, maybe that's not the sort of thing we need for some general applications. All right, so snake bite. I want to neutralize the toxins in a snake bite. Do I want to use a monoclonal antibody for that? Turn to your neighbor. See if you can come up with a response for that.
Well, so if it's a venom thing, you want to use something like fiber.
I'm not hearing much discussion here.
So I guess
Try. Talk.
Do you think that? So we're just giving it. And also,
It's what slide am I on? Is this slide not in our PowerPoint?
they don't have
Yeah.
the
No, it's just
Yeah.
no, but usually there's questions in here.
Yeah, he
All
has
right,
to
I'm
answer
gonna
what they
ask Rockford about this.
I don't even
Hey.
see it.
Let's back up. What are the venomous snakes here in Illinois? I know, Chicago, we've got politicians, but out there in Rockford, you might get out in the countryside, okay? What kind of venomous snakes might you encounter?
I think I speak for all of us when we don't know.
Where's the Boy Scouts and the Girl Scouts when you need them? They know all of this stuff about poisonous snakes. No Boy Scouts, no Girl Scouts out there?
I'm not from Rockford, just because I'm here.
Oh my. I'm going to speak to the admissions committee. There's got obvious discrimination going on here against the scouting program.
Yeah.
Well, there's basically two venomous species of snakes here in Illinois. You got rattlesnakes and you got copperheads. Maybe down in the southern very tip of Illinois, you've also got water moccasins. Okay, those are the three biggies here in the United States. You get way down south, Florida, Texas, that kind of place. You could also encounter coral snakes. So those are four venomous snakes. There are subspecies of rattlesnakes too, so you can get into that if you are interested. Anyway, you just been bit by a timber rattler. You want to inject monoclonal antibody to neutralize it, or do you want a polyclonal preparation instead? Which one would work better? I guess that calls on some background knowledge that I assumed you all had. But not being Boy Scouts or Girl Scouts, you didn't know that snake venom is actually a mixture of a whole bunch of different enzymes. Clots your blood, sometimes it attacks your nervous system, degrades tissues. Okay, so there's proteases in there and so on. It's a whole bunch of different enzymes. So again, monoclonal antibody, just one kind of antibody, or a polyclonal. Polyclonal. Very good. You'll live.
Microphone works. I checked it just.
Why?
I just have no clue. That's why.
You're on the right track. We're going to have to recognize a whole bunch of different kinds of toxic proteins. If I had a monoclonal, it might pick out one of those, attach to it, and neutralize it, but leave the others alone. They're highly specific. And so, yeah, maybe you don't die of the neurotoxin, but your blood clots and your tissues turn black and so on, and you die anyway. On the other hand, if you had a polyclonal serum, for example, by taking rattlesnake venom, diluting it down, injecting it into the horse over there on the farm, maintained by Eli Lilly or whoever's preparing this stuff, and then extracting the antibodies from that, you'd have a whole range of antibodies in that polyclonal serum, ones that would go after the blood clotting stuff, the tissue degradation stuff, the stuff that goes after choline and acetylcholine. Yeah, you'd block all of those kinds of toxins. So, given the choice, I'm going for the polyclonal, not the monoclonal in this case. This is an advantage of having polyclonal, a disadvantage for the monoclonal in this kind of application. All right, another one here. So you're running a drug screening program for addicts here. And you're trying to track whether or not they've relapsed and gone back to using hydrocodone. Would you use a monoclonal here to detect relapsing?
We're just looking for hydrocodone.
They can't get any, or maybe
They come into the clinic, you do a blood draw, you want to test it. They're saying, oh, I'm clean, I've been clean. I'm clean. Which kind do you want?
Good product. Just because the last one was
No, mine, because you were asking for
Mono because it's you making it come
What happens to hydrocodone in your body?
I don't know why I'm talking about it.
It's,
You're right.
what is it?
It gets metabolized. Yeah, considered metabolized here. You guys had 422. What kind of metabolites would be common?
Funny.
What? Hydrocodone.
Or get off. You would break it down to one hydro and one codone.
No, it's converted into morphine, right?
They were just a couple words.
Please, someone type it in.
Okay, in Chicago, I've got a hand up.
Yeah, but I don't know what hydrocodone.
Hydro.
It should be similar. It's similar effect. That's the only way.
It probably just has.
So the suggestion in Chicago is go for the monoclonal. Now that monoclonal would be for hydrocodone, I will assume. Are you going to pick up metabolites with it? Maybe, maybe not. Depends.
You're not.
All right, let's review. What kind of metabolites would you get? Well, in oxidized form. Probably the major route by which this would be excreted is they'd attach your body, your liver, would attach sugars to it. It'd be glucuronidated. Remember glucuronic acid? 422, yeah. So would the glucuronic acid get in the way of being in the site that would normally be recognized, the epitope, by your monoclonal? Maybe yes, maybe no. It would depend. You could have a monoclonal that would recognize the back end of the molecule, away from the site of glucuronidation, in which case, yeah, you'll pick up both hydrocodone and the metabolite. But depends. Which batch of monoclonals do you use? Did I recognize the site that gets glucuronidated? In which case, that sugar in the way might block the assay. And so you would not be able to detect that this person claiming that they're clean, they actually did use it, 2 days ago, and they still got some working around. Okay? All right, on to assays here. And this is the part that I suggested, again, last time, that really is confusing to students. So I'm going back over it again here. So, in a competition type assay, immunoassay, we'll use a heterogeneous format. Remember, that means that we've got a solid phase and we've attached stuff to the solid phase here. So we've attached antibody to it and it recognizes this analyte, and that could be drug, peptide, something like that. And we've got a labeled form prepared by us, by the boys in the back lab, that's the same as this analyte here, drawn from your patient or whatever, but it's got a tag on it of some sort. Could be radioactive, could be fluorescent, could be an enzyme. So what we'll do is we'll let this equilibrate, our equilibrium being established, and then we'll pour off the stuff that didn't stick. So that goes away. And what's left is what's stuck. And we'll look at that plate and see a signal. Now, how much signal do we get? It depends on, well, of course, how much of this labeled stuff we've got, but also, really critically, on how much of this analyte there is in your sample from your patient. If you've got a lot of this unlabeled stuff, chances are real good that most of the time it's going to beat out the labeled stuff. It's going to outcompete it. Law of mass action here. So relatively little label would get attached, and most of the binding sites would be occupied by stuff that isn't labeled. Do you see these ones that are occupied by the unlabeled stuff? No, no signal from them. Only if you've got the label bound do you get a signal. So you've got a lot of unlabeled. You got a lot of unlabeled here. Very little label. Very little signal, even though you've got a lot of the analyte. That seems to be the sticky point where people get confused. Conversely, if I had very little of the unlabeled, most of these complexes then are going to involve the antibody recognizing the stuff with the signal generator. So most of these things are going to have little signals coming out. So a little bit of analyte means lots of signal. So we're up in this region. So this is the one that's really hard to understand. Why don't we get more signal with more analyte? No, it's competition. And it drops as we add more analyte. Now, the converse here is what the book calls an immunometric format. I'm not entirely happy with that terminology, but it seems to be the one the industry uses. Okay, so in this case, let's make a sandwich assay, okay? And what we'll do is we'll put the analyte in, and we'll have an antibody that's labeled, and we'll have antibody on the solid phase, and it's a different kind of antibody than this one. And so it looks at different chemical aspects, different epitopes on our analyte. These guys look for the pointy end. These others that are free-floating look for the indentation end. So we form so-called sandwiches. Okay? So if we have a lot of this stuff, we can get lots of cross-linking here, lots of sandwiches, and we get lots of signal. What if we don't have very much of this? There isn't much analyte. Are we going to form many sandwiches? No. Little signal then. So the amount of signal goes up the more that we can form sandwiches, and that goes up as the more analyte we have. So the curve goes up. Signal intensity rises with analyte concentration. So we got that clear now. And this makes a really nice short essay question on an exam. Explain why the signal intensity drops in a competition assay. Explain why the signal intensity rises in the immunometric format. Can you do that? All right, so suppose you want to do an immunoassay on patient serum samples to detect antibodies to herpes simplex virus C. You want to use microtiter plates. It's going to have antibodies or whatever stuck on their surface, and either the EMIT or the ELISA format. And we covered that in lecture. There are two different kinds. They use enzyme. So, turn to your neighbors and quickly come up with a choice of which technique you want to use, and see if you can come up with a general explanation for why. We'll give you 2 or 3 minutes for that.
Oh, you do like a software, right?
Yeah. Not looking too good for him.
What do you think, Kurt?
I don't know what you mean. So.
By the way, I have asked exactly that sort of a question on exams in the past. Those of you that have looked at the old exams will have seen something very similar to this.
Yeah.
I'm hearing a little discussion here, but not enough. Talk, talk, talk.
It's just too many.
I just don't like that the numbers aren't in order.
It's all for the ring.
Well, no, it's easier at this than
Oh, except we can take this way. Follow him up.
Okay, we have a question in Chicago.
321. 11.
I'm sorry. Let me step away from the podium here. Why would I ask a question like this? Let's go metaphysical for a moment.
I just went through the
Why?
test already.
What does this
What?
contribute to your career at Walgreens? Well, first of all, remember a lot of point of care assays and home assays use EMIT, not so much ELISA. Okay, so that's kind of important. You may get questions from people about, can I test my urine? Can I test my kid for whether he's smoking weed? That sort of thing. How does that test work? So, yeah, you're the pharmacist, you're the chemical expert. You ought to have that background to be able to do that sort of thing. If you're working in a hospital, on the other hand, maybe it makes a difference as to, cost, how much these things cost. Are we going to standardize on the EMIT assay? Are we going to standardize on the ELISA for this or that analyte here? How do they work? How reliable are they? So you've got to know that sort of thing. All right, so here's a review. Remember how EMIT works? All right, we take an enzyme and we conjugate, like chemically, our antigen, our analyte, onto it. And we give it substrate, and that enzyme will go to work and cleave it and give us a signal. But we'll add antibody. Now, if the antibody's around here and it finds that antigen, it can stick to it, cover up the active site, and then the enzyme can't work. Or maybe it sticks to the antigen and causes the enzyme to change its shape. Compare the shape, rounded, squared off. And again, the enzyme doesn't work. Conformational change or steric hindrance here. All right, so what happens if we've got some exogenous antigen? Not shown on the diagram here. Well, that's free floating. It's going to stick on the antibody too. And if there's a lot of it around, what happens? Competition here. So the analyte with this antigen will stick mostly to these antibodies. And we'll see this thing stay intact. It won't get bound onto this because the antibody is not looking at this, it's looking at those free-floating antigens instead, holding on to them. So the enzyme stays happy and goes to work on your substrate and you get lots of signal at low free antigen. Or sorry, at high free antigen. By mistake, high free antigen. What if we don't have much antigen around floating in our sample? The only antigen around maybe is the one that's conjugated. What happens to our antibodies then? Well, then they're going to stick onto these, because those are the only ones available. They're going to shut down the enzyme. We won't get any signal. Pretty simple design. Let's go over the ELISA. All right, so there's two formats. I think the sandwich one is a little bit easier to understand. But we'll have our coated plate here with antibodies, monoclonal perhaps, okay? And we'll add our antigen and it sticks, and then we'll add a secondary antibody with an enzyme hooked onto it, enzyme linked, and that forms the sandwich. We've shown that picture before, and now we get lots of signal out here if we form lots of sandwiches That is, if we have lots of antigen. If we don't have much antigen, not so much sandwich, not so much signal. What are your advantages for one versus the other? Which one would work for really small drugs? Which one would work for big things like, well, a hepatitis C viral coat protein? Which one can we get away without using monoclonals? Do we have to have a monoclonal up here? No. I can use polyclonals. They just recognize different aspects of the same antigen. Fine. Cheaper. Gotta use monoclonals for these. Expensive. If I want to be exquisitely sensitive to one particular strain of hepatitis C, yeah, maybe I don't want to use this. Maybe I want to use an ELISA that would have antibodies that would recognize that particular variety of viral coprotein and no other. Okay, so summary here. Which format's better? Well, both are using enzymes, both use antibodies here, but EMIT, we can get away with only one species of antibody, and in fact, we can often just use polyclonals there. Whereas the ELISA calls for at least 2 different species of antibody, and usually they're monoclonals. Okay? Now remember, for ELISA to work, we've got that sandwich format. We've got to have an antigen that's got a front side and a back side that are different enough so that we can get antibodies that are different and recognize those fronts or backs so we can form the sandwich. We don't have to worry about that for EMIT. So that's a problem. Now, the EMIT is nice and homogeneous. One pot holds everything. Don't need to pour and separate. Here, yeah, a couple of wash steps, 1, 2, 3 wash steps in order to get to that. More handling, more technicians, more expense. So EMIT's starting to look a little better here. All right. With ELISA, you also have to do a bunch of linking here, which you don't necessarily have to worry about with the EMIT. So on the whole here, I'd probably go with the EMIT format over the ELISA. We really ought to have some customer rep from a company that makes ELISAs up here to debate me on that one, though, for fairness. Question in Chicago. So if we were trying to detect a very specific strain of virus or bacterium or whatever that I would want to use ELISA, I would say that ELISA gains a bit there if I'm trying to discriminate that strain from any other strain, if I've got to do a differential diagnosis. If it makes a difference. On the other hand, if I just want to say, yeah, you got Hep C, maybe the EMIT's a better choice. Does everybody follow that reasoning? I hope. Okay. All right, so things can go wrong with immunoassays. So one of the most common things that happens here is that you've got a blood draw from your patient. Maybe they clear it, okay? So it's now serum, so there's no blood cells and no platelets, okay? But there could be soluble, free-floating stuff still in your sample, and would it interfere with your enzyme? Could it inhibit the enzyme? For example, maybe they've got a lot of phosphate in their blood. Well, it turns out that alkaline phosphatase, which is a common enzyme used for these EMITs and ELISAs, Yeah, that gets inhibited by phosphate itself. Maybe they've been treated, the technician that did the blood draw added some EDTA to the vacutainer. Everybody knows what a vacutainer is, right? You've all had blood draws. So they added some EDTA. Now, why would they do that? Well, it chelates magnesium and calcium, keeps things from clotting right away. Maybe they wanted to preserve that. And you got the tube to analyze. Oops, it's got EDTA in it. And EDTA will not only chelate calcium and magnesium, but it'll chelate zinc just as well. And as it turns out, again, you got to have some zinc around for that enzyme to work. Oh, that would shut down any signal generation. You've inhibited the enzyme. Horseradish peroxidase, same kind of problem. You got any chelators around for calcium? It's going to interfere with that. There are other possibilities here for enzyme inhibition as well. You might get some sort of background signal. Let's not use enzymes. Let's just tag things with something that fluoresces or that has really good UV absorbance so I can see it. Well, as it turns out, for fluorescence, nicotinamide fluoresces real well, but so does bilirubin, and that's a very common thing to show up in blood draws. Okay, hemoglobin absorbs in the visible in the ultraviolet, so that would block the generation of any fluorescence. And then there's vitamins. Go back and look at the structure, for example, of riboflavin. You got a big conjugated pi system there, and that absorbs in the visible region very broadly. So it's going to interfere again with generating any sort of a signal, spectroscopic signal. This is a repeat of a slide from the previous lecture, so I'm not going to go over it again, but we've just looked at endogenous signal generating substances here, or enzyme inhibitors, or contaminants, and so on. But there's lots of other things that can go wrong as well, and I did cover a number of those in the lecture, so please review that. All right, I think it's time to take a 5-minute break. It's 10:25. Let's come back again at 10:30, and we'll carry on from there.
Hey, are you memorizing dosages?
Just for levothyroxine.
That's it?
Yeah.
Not for
Any other dosage?
like PT or methimazole?
I mean, like, he's not gonna ask us to go dose this.
Like the average dose, like 15 to 60 hundreds a what?
Look, it's gone.
Yeah.
I don't know. There's no coming back to class over.
I know, like, the only doses he specifically talked about was like T4 and T3 doses. Those were what I
Yeah, you
expect
really got sick.
to be
All
hard.
for
How was
thyroid
I
stuff.
Thyroid stuff, I know, but I didn't look at the renal dosages because I don't think it were such a big deal.
Yeah, I think with the levothyroxine, it's one start bottle, otherwise
It depends on the populations though.
Population stuff. Like as he said,
Like
like, depending on
geriatric.
the population, you should be able to identify the correct dosage. I don't want you to know the exact thing, but you want to identify. I don't know about adrenal if he said anything like that.
Your voice is gone. Yeah.
Well, adrenal is like the long acting, short acting, intermediate, and long. That stuff, right?
Throat.
I think so, yeah.
No, yeah, like the betamethasone, dexamethasone,
Maybe you have allergies.
triamcinolone, prednisone, hydrocortisone, cortisone.
So
I feel like
you
once I'm
think
done
it's
taking
a
this
cold
test, it's just gonna all
Well, you-
like
Oh, wait, 100%. Dude.
Essentially, yeah, that's pretty much it.
I'm ready to take it right now, dude. Like,
Honestly, the thing
Yeah.
is, the margin of error is such a small thing. That's the issue. Otherwise, it would have been fine. 23
Yeah.
not
questions.
enough questions,
Like,
yeah.
come on.
Usually I'd be down for less, but
Because 23 questions, you usually overthink the hell out of it.
23 questions?
Yeah.
That's it?
That's
yeah.
exactly.
That's a lot of information for 23 questions. Like,
I think a
the
lot
thing
of them are gonna be like those
It's not going to be
cases.
patient cases.
Yeah, it's going to be like, okay, what's the possibility of this patient getting Graves'
Oh,
disease
we have an
with
exam on
tumor
Monday.
or something?
Oh.
That's going to be the answer.
I think a lot of
That's
it's gonna
also-
be diagnostic too.
Just assume that we don't diagnose anything, but
That's what he likes, so.
Oh, cool. Okay.
We have so much.
oh, you were playing the shit. Oh man,
Didn't realize you were here, Mark.
Oh yeah.
Hiding your bag.
send me the link.
I got you.
Stealth mode.
Yeah.
Just send me the video.
Yeah.
It took me an hour to get from
That's a good idea, actually.
Yeah, just don't like tell everybody though. It's like illegal.
I got it on Amazon. It was like, I remember like a few years back. I'm like, when the hell am I gonna ever need it? But I can't.
You sure? You sure?
Integrity.
I know.
Because he's given us basically test questions.
Keep on download.
Taking evidence right there. Should report you or else I'm dishonest.
Exactly.
Show Kurt how to hop out of stealth mode real quick.
Exactly.
You must be red.
I did a lot of buffalo balls yesterday. Some KP work.
One to
And then it was like, you
colors.
guys want to continue doing KP work and but not gonna study for my exam,
That shit
thank
is
you.
like-
But yeah, it's
Oh.
only like
Oh no, I just connected it. Yeah.
The night
so you take something now, you put something yesterday. Feels though, it's just body trying to find something. You should cover your chest. Don't get burned out. Just wait.
Don't do anything.
Okay, we've got a clinical case here. This one came from Great Britain. A real case. So this 52-year-old woman who had been previously treated for irritable bowel syndrome, they checked out her hormones and they found a number of gut hormones were elevated here. Now, even though the imaging stuff doesn't show any real abnormalities, maybe a little bit of growth in the pancreatic islet cells, but nothing unusual there. She's not receiving any drugs except that from her estrogen implant. So they think there's something wrong here. They want to go in and examine that pancreas in some detail. So pancreatic angiography, which is pretty invasive. They got to thread, open up your belly, go in and thread stuff into the pancreas, which is a very delicate organ as well. As it turns out, she and her husband were rabbit raisers. They keep rabbits. And I know that's not so popular as a food here, but in Britain, apparently, they eat a lot of rabbit. Okay. So they keep them around, and she had complained that, yeah, I'm kind of allergic to rabbits. My nose runs a lot, and I sneeze. All right, so some questions for you here. Why do we see such a high level of gut hormones in this case? What's going on here? Notice that these aren't necessarily all from the pancreas. Yeah, glucagon. But does gastrin come from there? No, you might want to check that out. Where does gastrin come from? In VIP. Somatostatin, yeah, that comes from there. But somatostatin has another source as well. So it's pretty unusual to see more than one hormone rise if you've got, say, something wrong with one of the endocrine cell lines. Sure, if you had alpha cells that were going nuts, so yeah, you'd produce lots of glucagon, but you wouldn't necessarily see a rise in these others. So what's going on here? That's one of the questions, of course, that they want to look at her pancreas to try to answer. Now, here's another hint. Rabbits, in addition to pigs, sheep, etc., are often used as a source for antibodies that are used in diagnostic tests. So what could be going on? All right, so this is the kind of assay that they would use to analyze this. So what would the analytes be here? Well, it would be all those peptide hormones. So we'd have antibodies, presumably from a rabbit, here. And here would be free-floating hormone, and we'd have laboratory-prepared labeled hormone, and we'd do a competition here, and we'd see our usual competition sort of format, drop off in signal as we have more of that stuff. So that's the reading that you get if there's a lot of free-floating hormone. You don't see much signal coming out. All right, so if those antibodies are from rabbits, what could be going wrong? Well, the MD who saw this is actually the second MD, I think. Before she went in for surgery, said, let's do another test. Let's take that assay buffer. We were assaying for gastrin, one of the hormones here, and let's add some non-immune rabbit serum to that. They went, whoa, things go down. We're back to normal here. Well, all right, so the upshot of this is the patient is producing antibodies to rabbit stuff. Okay? I said rabbit antibodies, but it's antibodies to rabbit things, okay? And that's what's going on. It's going to interfere with the various gut hormone assays. The readings that without the serum were high. That is, readings that indicated a large amount of hormone. And these antibodies from the patient were there in the sample. So if we back up, not only do we have these antibodies, ones we've got on the plate, but we got some free-floating antibodies from the patient as well, from a blood draw. And if they're going after stuff, well, if that's an antibody from a rabbit, and the antibodies we just added look for rabbit antibodies, what's going to happen? It's going to stick on here. Okay? Yeah, it's going to compete. All right, so, all right, I can see that this is getting a little bit involved for you. Well, the upshot of this is that because we've got competition here, the gastrin was blocked from binding.
Recitation.
All right, so it remained free, and so it appeared to be in higher concentration. And so if they weren't raising rabbits, we would have not seen this at all. We wouldn't have had this interference from the patient's own anti-rabbit antibodies. So that's just one more source of interference here that's possible. Well, when they go back and they check it, again, by adding non-immune rabbit serum, that brings things down. And thank goodness the patient was not producing abnormal amounts of all of these hormones after all. And that saved her an awful lot of problems. Okay, so I will put up, I guess I will, yes, put up all of these slides for you to review so you can try to follow the logic again. All right, what about those urinary drug screens? Well, we talked about hydrocodone, okay? So opioids here. There's lots of things that can interfere with opioids. So I think people have heard about poppy seeds. If you're in a drug screening program, don't eat the bagels with the poppy seeds. You'll test positive for opiates. All right, what about methadone? Well, as it turns out, diphenhydramine. Everybody knows what that is?
I don't know.
Yeah. Antihistamine, very common antihistamine here, okay? It'll interfere with that, give you a positive result. Okay, benzodiazepines, barbiturates. Barbiturates, people are taking ibuprofen by the handful. Are they really abusing barbiturates? Yeah. Okay, so there's a lot of false positives out there. Now, metabolites, as we've already talked about, could produce glucuronide metabolites, and we've got other kinds of metabolites when we hydroxylate things. All of these could interfere or be registered as positive, or generally interfere, I guess I should say, with your assay. A few words about labels. So we've talked about ELISA, we've talked about EMIT, and those use enzymes here. So the most common ones are going to be stuff like horseradish peroxidase, alkaline phosphatase, and glucose-6-phosphate dehydrogenase. It does complicate things because you've got to add substrate here. And that can get you into some difficulties also with trying to conjugate things. Wouldn't it be simpler to tag things with a single atom of radioactive stuff? Well, that was the original format, remember, for Roswell Yellow for checking for insulin. So she tagged things with I-125. And it's possible to do other kinds of radioisotope labels as well. But because of all the hassles dealing with radioactivity and the fact that a lot of these other assays have now gotten very sensitive, this isn't seen very much. It's still out there, but it's generally relatively specialized. Sort of in between is adding something like a fluorescein or maybe another kind of fluorophore here. And that can be okay, but again, background signals can be a problem, give you interference. All right, so another discussion problem. Why are bigger antigens better targets for labeling? And what are some disadvantages of labeling the antigen? What about attaching a label to the antibody? Why would that be a good idea? Remember how labels could go on to an antibody. What assay format does that lend itself to? Can you recall the different assays? Come up with answers for those quickly. Well, let's take the first one. What about big antigens versus little ones? Isn't bigger always better? Bigger antigens are going to give you more of a place to attach things, okay? And you can add different kinds of labels here. You got a possible range of choices there. Also, with a big antigen here, you could have front and back sides, so they could be separated, your label, from what's recognized by the antibody, so it won't interfere. Now, maybe that antigen doesn't have a variety of attachment points, particularly for small ones. So if we're trying to label Celebrex, for example, yeah, a problem there. Antibodies are pretty big. Well, again, for example, we could put our label maybe down there on the FC region, the stem of the Y, and that would be far away from those antigen binding sites. We could hope then it wouldn't interfere with it, okay? Also, again, more possible types of labels. I could use radioactivity, I could use fluorescence, I could add an enzyme on there. All right, so cathinone is the active ingredient in cotton, which is this shrub whose leaves are chewed by people living over there in Eastern Africa and parts of the Middle East, okay? Cathinone looks an awful lot like amphetamine and produces pretty much the same sort of effects in stimulating the central nervous system. All right, so come up with an immunoassay for cathinone. You want to do EMIT, ELISA, you want to do competition, you want to do immunometric. I'm going to leave that as sort of a homework problem for you. Okay, so think about what criteria you would want to do. How big is this molecule? Can we attach it? Can we label it? Or do we want to label antibodies? Do we want to take this and stick it onto a big enzyme itself, like for an EMIT assay, and so on. All of those are sort of design considerations here. Then, what's going to get in the way? What sort of interferences? Well, clearly amphetamines are going to interfere, but would there be other things that could? Let's see, amphetamines. Oh, there's a whole bunch of things that can give false positives here. Okay, so like I said, I will post the slide here with this sort of general discussion. It is not a black and white thing. This is real life. This is the sort of discussion that you would have with colleagues if you were trying to work for a pharmaceutical company and develop these sorts of assays. All right, so we talked a little bit about hep C. Let's talk about hep A and detecting it. Okay, this stuff is pretty contagious. So once it gets started, say in a college dormitory, yeah, you got a problem. It's transmitted oral fecal route, as I recall. Yeah. All right, so let's think about an assay for this. So step one, we'll take patient, we'll take antibodies from it, take their antibodies from the patient, we'll put them onto the surface of our wells of our microtiter plate here. We'll add viral antigen, and of course that'll get captured by our patient's antibodies. Then we'll add immunoglobulin G and immunoglobulin M from their patient's free-floating ones here, represented by these blobby things. And you'll learn more about those structures next year. And let them equilibrate, so they'll form some sort of a sandwich like that. And then third, we'll add a labeled anti-HAV immunoglobulin. And so what that will do is stick on there. Now, it'll presumably also have a label on it and get either a chromophore or a fluorophore on it, okay? So we would see this sort of triple sandwich sort of thing. Now, would this work if we were going to use it to, we've got a whole dormitory of students here, and we want to test them to see if they're going to come down with hepatitis. What if they've been immunized previously? How would that interfere here? Is this going to work? Well, would they have antibodies to hepatitis A if they've been immunized? Yes, they would. Just like they've been infected. Only they're not producing virus, presumably, but they'll still have circulating antibodies. They'll probably be immunoglobulin G and immunoglobulin M. Oh, wait a minute. Isn't that just what we had here? So we'll end up forming sandwiches. Ooh, this will give you a positive result for those that have already been immunized. And it's not a good way to discriminate people that have just been infected by the virus versus those that were already immunized a while back. Okay, let's talk briefly about Western blots. So I think everybody was cool with electrophoresis to separate proteins. Were there any questions about that? So we take the gel that we've run the proteins out on, and we blot it. And that, through some magical chemical process, the proteins get blotted onto the special receptive paper, nitrocellulose or some sort of polymer that sticks them on there really well. Usually nitrocellulose, but there are others available. And then we bathe it, say, with HIV antibodies, and we incubate them with enzyme-labeled antibodies that recognize that FC region of these HIV antibodies. So if any of these stuck to proteins from the sample, we're going to get a signal from those enzymes in this sandwich. Okay, get a color development. So let's go back over it again. We electrophorese here. There may be HIV proteins in there. Okay, we blot it. Okay, it goes on to nitrocellulose. We add antibodies. Now the antibodies recognize this, that, and the other kind of protein. And then we bring in a second antibody that's got an enzyme on it. And that recognizes, say, the FC region of these antibodies. These primary antibodies only stick to those bands that were from HIV. They don't stick anywhere else. The secondary antibodies don't stick anywhere but to antibodies that are already there. So we'll see enzymes stuck there, and we'll add substrate, and we'll get color, da-da-da-da, right where those are. So that's the Western blot, and that's how you could do it. Instead of doing an ELISA or an EMIT, you can have all those college students in the dormitory give a donation of their blood. You'll do a Western blot. One of the advantages here is that we can actually run a whole bunch of samples in parallel. Those EMITs, those ELISAs, it's one pot, one signal. If we do electrophoresis, I can electrophoresis student A sample next to student B, student C, D, E, F, G, all the way across. So I can actually analyze a whole bunch at the same time. That might speed things up, save some money too. Just a point of comparison, we're forming sandwiches here, but in this case, okay, we're forming it with antibody to antibody. Look, it's recognizing the FC region here. That's how that worked. This would have been our nitrocellulose over here. These would have been those HIV proteins on the surface that we'd stuck from the gel. They'd stuck onto the nitrocellulose. So why do we use primary and secondary antibodies? Why don't we just label that primary antibody and forget about the second one? Well, it's cost, partly. That primary antibody is supposed to recognize only HIV and nothing else, which means we really want to have stuff that's pretty specific. How do we get specificity? Monoclonals. The polyclonal stuff, not so specific. Okay, so you probably wouldn't want to choose them for that. Unfortunately, monoclonals cost a lot. Furthermore, if I labeled it, it might interfere with its ability to recognize the stuff that's on the surface of the nitrocellulose. It wouldn't recognize the HIV proteins, maybe. Problem. Why waste all that money and get a null result then if it doesn't stick? The secondary antibody, the one with the label, those can be anything that recognizes FC regions. Yeah, we don't need to have anything that's real specific. It can be a polyclonal. And it can go after all sorts of antibodies, just as long as they've got an FC region. We'll recognize it with these antibodies. And that way we can lower the cost and make the assay cheaper overall. And we'll skip that. All right, so we've talked about why RIAs aren't used so much. Here's a couple more reasons. So you'll want to review that because, yes, another exam question is, why don't we all use RIAs anymore? Well, yeah, radiation is a big hassle. Here's another problem, though, that half-life of the isotopes. Well, some isotopes last a long time. Tritium, half-life of 11 or 12 years, fine. Iodine 125, 2 weeks, maybe, something like that. Iodine 131 is a couple of days. So that means, 3 days later, you only have half the radioactivity that you had. Your assay sensitivity is dropped by a factor of 2. Oops, not so good. Well, now, this, again, was a basis for an exam question from years past, not in this course, but I think in a previous course. So ACTH, adrenocorticotropic hormone, 39 amino acids, big, 4,500. It's got tyrosine in there, so we can label it by 125. And you want to do either an RIA or an IRMA. Oh, yeah, IRMA. Immunoradiometric assay. You have to review your slides. Which one do you want to do? And draw a sketch. Represent the assay. And are you going to see more signal with more analyte, or are you going to see less signal with more analyte? Which way does the curve go? So a homework problem there. Again, this is the kind of open-ended thing that has no one single right answer to it. You have to justify things along the way. So here's just a review slide, and again, I will post these for you so that you can look at it. And for RIA, this is what the curve would look like, okay? So you'd have a standard curve where somebody in the laboratory had taken known amounts of ACTH and seen how much signal you'd get, and this would drop off like so. And if your unknown sample is out here giving you a reading like that, you can figure out how much that unknown ACTH is. All right, we're almost done. We may finish a little early here. Let's talk about agglutination type assays here. So the precipitation is when we've got small soluble bodies that are getting clumped together, and agglutination is when we've got big things being clumped together, like red blood cells. So this shows up probably most commonly in blood typing, but also in those bead-based point of care tests at home diagnostics. So let's take the example of looking again for HIV. So we've got red blood cells, and they've got a surface antigen, that probably is glycophorin. That's a little spiky triangle there. We have a patient blood sample. We think that they probably have HIV present. Well, they probably then raised antibodies against proteins from HIV. So in their blood, they'll have regular red blood cells, and they'll have free-floating antibodies whose FAB regions could recognize a protein from HIV. So what we'll do is we will add to this preparation a secondary antibody. Now this one, we've chemically attached a fragment of an HIV protein onto it. So we'll call that the antigen here. Okay, so it's chemically linked. What is this thing going to recognize? Well, we've got this and this that recognize this. What does this recognize? Does it recognize that? No. That recognizes something from HIV. Oh, that's this stuff. Is everybody clear on that? So these are entirely different specificities. This laboratory prepared antibody that has chemically linked onto it stuff from HIV will look at and bind to red blood cells, to that glycophorin. The patient is producing free-floating antibodies against, well, HIV proteins, just like this stuff that we've chemically attached. So we mix things together, and what's going to happen? Well, these guys like to stick to those little triangles. So here we have a red blood cell, and here's our antibody sticking to it. But if there's this kind of antibody from the patient present, what does it stick to? Why, it sticks to those antigens there. And look, because of that hinge region and flexibility, I can cross-link this red blood cell to this red blood cell. And maybe I bind a different one over here, and this could cross-link to yet a third red blood cell, and a fourth, and a fifth, and a seventh, and 20, and I got a big clump after a while. All right, so is this a precipitation assay or an agglutination assay? Everybody wants to leave early, right? Responses from the audience are desired so that we can all leave early. It's agglutination. This is a big article here that's being clumped. It's a whole cell, not a small drug or a peptide. So it's an agglutination assay. And if you weren't sure about that, I gave you a big clue right here. Okay, we're almost done here. I think that the fluorescence polarization immunoassay stuff I went over too quickly, so we'll do a little bit more of this. This actually gets used here in our university quite a bit, so it's worth talking about. This is the kind of thing that often shows up in drug discovery programs. All right, so what we're going to do is we're going to shine some polarized light at our sample. And it's going to absorb that light, and then it's going to fluoresce. And when it emits the fluorescence, the plane of the emitted radiation will, we hope, sort of match the incoming plane of radiation. And that's what we mean by polarized light. Wow. But sometimes these things wait a little bit before they emit their radiation. If they rotate a little bit, now they've changed their angle of emission. If they're really small, they might have rotated quite a bit. If they're really big, because of bigger frictional drag, they won't have spun around quite as much. So we'll measure whether we've got really a little bit of fluorescence coming out of it that's polarized, or a lot that's coming out polarized. If it's fluorescence that's really polarized, then it's big, whatever it was that was emitting. High molecular weight, lots of drag. If there's very little polarization, not fluorescence, it can still fluoresce, it just doesn't orient itself the same way. If it's at all different angles, then it was a much smaller emitter. All right, so what we'll do is we'll take our analyte here, and we'll have some authentically lab-prepared analyte that's got a fluorescent tag on it. And here's the antibody. So we want to see if this stuff is around or not. So we're going to set up some competition here already. The antibody detects these and binds them. If there's an overwhelming amount of this, yeah, we got competition, so we won't get much signal. Well, we'll tune it so that we add an appropriate amount of these, so that we do get some fluorescence. Well, now, this thing is bound. It's got the fluor on there. It's bound to this humongous protein, 150,000 molecular weight, typical antibody. That's much bigger than these small antigens. So we look at the fluorescence polarization. Does it stay polarized? Then we had a lot of this binding. Well, if this bound a lot, then chances are good it was really out competing these guys, so there wasn't much of that around. If there was an awful lot of this, what would happen? Then these guys get out competed, and we don't get binding to the antibody of these labeled antigens or analytes. We get mostly this kind of species instead. Are they going to fluoresce? No, they won't. What about these free-floating guys? Well, they can fluoresce, but they're small. They spin around a lot. Their fluorescence won't be polarized. Yeah, we'll get fluorescence from this and from this. What's the proportion that stays polarized? That's what we're looking at here. It's not whether we've got fluorescence. It's degree of polarization. So, when do we get lots of polarization, remaining polarized? When there's very little of this. And mostly our label gets stuck on the antibody. All right, so here's a typical small molecule. It'd be hard to get antibodies to it, actually. Gentamicin. This thing is not very big. It could be hydrolyzed, but it's not chemically distinct enough. It's got amino sugars, but really doesn't look a lot like it. So let's hang a tag on it. All right, so we make gentamicin plus fluorescein. We'll test it against free-floating gentamicin. We want to make sure that we're giving the patient the right dose and not getting too much antibiotic. We'll test it with these antibodies. All right, so we'll use fluorescent polarization. Suppose the patient had a overdose of gentamicin, that could be really toxic for them. Would we see a lot of polarization in our assay then? Yes or no? No. Why is that? If they've got an overdose, they've got an awful lot of free-floating gentamicin. That's going to outcompete our labeled gentamicin. So we'll have in solution mostly complexes like this, and most of our label is this. Is this stuff going to give me a nice polarization signal? No. It's small. It rotates. It emits the fluorescence at all angles. What if they have an underdose of gentamicin? What would happen then? Well, we still have competition, but now we'd be forming more and more and more of these kinds of complexes that are fluorescing, but they're not reorienting, they're not diffusing rotationally, and so their polarization stays the same. We get a high polarization signal out. Okay. Yeah, we could have problems with those other aminoglycosides. We could have cross-reactivity, so I just thought I'd throw that in there too. But structurally unrelated antibiotics probably wouldn't interfere with our assay for gentamicin. Okay, for your homework, think about, are you going to have problems with cross-reactivity, particularly with ELISA, with these kinds of drugs? What about there's a whole bunch of different anti-seizure drugs. A patient often is on more than one of those. What about cholesterol-lowering drugs? Would you probably have problems with interference there? Well, generally speaking, probably not, because usually they're on just one, but not a whole bunch of different statins. But over here, yeah, it's not unusual to have people on both phenytoin and valproate at the same time. And maybe they've got one of these others added in there to control different kinds of seizures. Okay, that wraps it up for today. So we are going to get out a little bit early. All right, questions from Rockford? Questions in Chicago? All right, we're done.